Yeah, so the topic of my talk today is you can read the title in there, and it's I'm gonna give you an overview of some of the work that we've been doing in the lab, not just what we're doing currently, like a timeline of how things developed here. And even though this is a very fancy and long title, basically what we are trying to do is can my dental feeling last longer, right? That's what this is what it boils down to. The science is cool, everything else is cool, but what we're trying to do is really help our patients. A little bit of a plug for my lab and us here at OHS2. We are located on the west coast of the United States, very close to Adriana, actually. We could drive. It's a long drive, but we could drive to Vancouver, BC. This is a snapshot of Portland, Oregon at night with one of its many bridges. It's a really beautiful town. Kind of outdated picture of my group many years ago. You see, I kept that one because Dr. Fogeling is there, and we're very proud of how high she went after leaving the group. The lab is here in this new facility, well, 10 years, 12 years old now, facility, now on the 7th floor of our LSP. And if you're interested in other projects and capabilities and things that we have in the lab that could be potential collaborations, please scan the QR code and get in touch. We'll be happy to talk to you. All right, so just to set the stage here, what we're talking about are restorations, right? Dental aesthetic restoration. So you know that when you have a lesion that is confined to enamel, that's a pretty easy thing to restore, pretty good prognosis, right? We can do this in our sleeps, everybody. We can teach our dental students. This is all great. When you start getting dentin involvement, but you still have enamel margins, that's still a good prognosis. The problem starts when you start getting endo involvement, and the size of the restoration that you have to place starts getting larger, and then the tooth might actually end up dying, and you either end up with having to put an implant in that tooth, which is you're getting less and less conservative as you go, or you might end up with an infection that can have actually systemic problems. So our job here as researchers, I think, for dental materials is to make the buck stop here. We want to create materials that the best thing actually before that would be that everybody spends $5 in a very fancy toothbrush and just brushes their teeth. And I would love to be out of a job, honestly. But that's not the reality. So if we have restorations, we want the buck to stop here. So you place the restoration, and it lasts for a long time, and at least the patient doesn't have to do retreatments. So very brief or overall view of how we look at this problem is if you look at the restoration, you can kind of didactically divide the culprits for restoration failure between the adhesive layer. So I'm talking about composite, the adhesive layer, and composite, the actual composite material. So if adhesive layer fails, you end up with either secondary decay or marginal staining. And secondary decay just means that you opened up a space for bacteria to recolonize. If the material is not strong enough, you might end up with fracture either of the material, or you might end up with fracture of the tooth because of polymerization stress. So some of the things that we've done in the lab on the composite side, one of them, very briefly, was develop this styrurethane additives. I call this vintage Pfeifer lab research. But it's a pretty interesting, of course, in my opinion, biased opinion, it's a pretty interesting additive because it's able to reduce the polymerization stress by up to 50%. And increase fracture toughness by twofold in some cases. And that comes without prejudice to the degree of conversion. So it was very exciting, something that we're trying to actually license into a material. The other side, on the adhesive side, this is actually thanks to a lot of work from Dr. Fogeling. We developed these multi-acrylamides, and those adhesives, because they lack ester bonds, they can withstand hydrolytic degradation even at very low pH. We tested this for 30 days at pH of 1, and the monomers stay intact, which is more than I thought would happen. They both increase the immediate bond strength, but importantly, they keep that bond strength over time. And that very tiny graph that you see on the bottom is just a snippet of all the microtensile bond strength sticks that Anna did while she was here. And we tested dozens of different compositions in different storage times and all of that. And then we came to one that's a multi-acrylamide that actually surpasses even the commercial control. which is pretty awesome. And you should hear us in the lab. We were screaming and jumping up and down when that happened. And this is all great. We're all very proud. This is awesome. But what is missing from that picture is that it does nothing to do with the biology of the oral cavity, right? So ongoing challenges that we were trying to address, and this is going to be the bulk of my talk, are things that are related to bacterial degradation and things that are related to host-derived enzyme degradation. So we're going to talk about biofilms, and we're going to talk about MMPs and how they degrade collagen. So that's to set the stage for what we're going to talk about. The other thing that I wanted to say is that we have to do a little bit of soul searching, I think, in the dental materials community and on how we test and how we approach those problems. Because I think we're past the time when you have to make the material stronger and just look at the material in isolation. So it's very important for us to start figuring out ways of measuring those materials in simulated conditions that are more relevant to the physiological environment. So Jack Farrakeen and I, both our mentor, long-time mentor, he is the big name, big shoes that we were trying to fill with everything that we do every day. He came up with this, what I'm going to call bioreactor for the rest of the presentation. And this is a clever way of coming up with a simulated restoration. So you basically take a disc of denting and use CNC milling to come up with a very standardized type of prep, which you then restore. That little puck here goes inside of this device, and this goes upside down. So the restoration side is looking down. There's a ring over here onto which that restoration sits, and then a piston that flexes the restoration from the top. So you're basically flexing the bonded interface as you go. So it's a mechanical cycling of the bonded interface. This is, as you can see, is inside of a chamber that allows for circulation of culture medium and also different gases. So you can have bacteria and mechanical loading at the same time. So Jack came up with that. I'll show you a picture of the patent that then led to a piece of equipment, and there's a disclosure when I get to this. But what Jack was able to show is that this is something that hasn't been shown before, is that when you look at the line angle of the restoration, so this being the denting and this being the composite here, denting and the composite, you see that everything that's in red here is bacteria. So the bacteria can actually not only penetrate, but stay viable all the way to the line angle. So this is a problem, right? This is a mechanistic study that shows how secondary caries actually develop. So after many years, and I got involved with in partnership with a Brazilian company, Odemi. I don't know if there's anybody from Odemi watching today. The disclosure that is missing from that slide is that we have a patent with them with OHSU. So there is a commercial product that comes from this. So this is my plug for the conflict of interest here. But we basically were able to take those chambers that I described in the previous slide that Jack developed and put this in series on a much more compact device that has a much smaller footprint and does basically the same thing. So you see, I'm not going to dwell too much into the description because I described this in the previous slide. So we're now having a higher throughput of those tests rather than having to tie up an MTS for an entire week. We can have this on a standalone machine and do 3 at a time. And then Fernanda Lucena, I don't know if she's here watching, but she was here in 2019 for her PhD sandwich coming from Brazil. So, smack in the middle of the pandemic is when she was doing all of this. She took our thiourethane composites and our acrylamide adhesives and combined them, and we measured them and we tested them in the bioreactor. So I'm not going to, spend too much time talking about the details here, but if you just concentrate on the bottom row here, that's just the bioreactor results. The commercial control is here, and then the actual experimental control is here. You can appreciate that there's a pretty big gap, a pretty big opening at the margin between the denting and the composite with the control, and even with the commercial control, whereas the thiourethane adhesive acrylamide adhesive combination completely kept it closed. And this is, the cycling that we do simulates 1 year in use in the oral cavity. That was another time when we were screaming and going up and down like crazy in the lab. We're very animated people in this lab. But this was exciting because you could see that this was a very well-preserved margin. But then we had to explain, so how did that happen, right? So how did that happen? So now then that's when Poliana Scaffa was here. I don't know if she's watching either, but she's at Unilever now, along with Steve Lewis and Matt Logan. So Steve is a mechanical engineer that's basically the boss of everybody in this lab. He knows all the equipment. He trains everybody. So if you want anything, Steve is your guy. And Matt is the chemist. Both of them have been in the lab with me for 8 years now. It's going to be 8 years and 2 months. So anyways, the three of them working together. And then there's some work that Anna did in the midst of this too. Is that there are too many pictures for me to put on that slide, but this is all part of Anna's paper. What you see here is remaining monomer as a function of time at a pH of 1 with the different acrylamides and HEMA. I mean, a picture speaks for itself, right? You have almost 80% degradation of the HEMA in that time at that acidic environment, and the acrylamides basically survived. And then you put those in storage with cholinesterase and pseudocholinesterase, and you'll see that whether HEMA degrades in 1 week by over 30%, the acrylamides stay intact. Then what we did was, okay, so we kind of knew this was expected, right? We kind of knew this was going to happen. It doesn't have esters in them, so this was expected. What was not expected is that we noticed also that those acrylamides were somehow reinforcing the collagen. And those two graphs are basically parts of the same story. This one is showing hydroxyproline concentration, so it's basically the amount of degradable byproducts of collagen. So the less you have here, the better preserved the collagen is. And this is shear modulus of the dentin itself that was stored with those adhesives. So without going too much into detail with this, what this is showing is that through some mechanism, those acrylamides are actually helping preserve the collagen. So we investigated and I'm gonna stop there because then there is a follow-up paper that came from that I'm not gonna have time to go over that showed that this is not because they're inhibiting MMP activity, but because they are doing some supramolecular interaction with the collagen fibers. So very interesting on that as well. We've also demonstrated that those adhesives so far, we had shown everything with the 5th generation adhesive. So you have the primer and the bond in the same bottle. Then Fernanda Suzuki, another current postdoc who was here did that work as part of her sandwich, the PhD sandwich program also. She demonstrated that technology can also work as a 6th generation adhesive. And again, in this case, she did a combination, different iterations of putting the acrylamide in the primer, putting the acrylamide in the bond, then combining a primer and the bond with the acrylamide or the control. And the overall conclusion with this is that the HEMA primers are good because they allow for some low viscosity and tags to be formed. But then when you combine this with the BAP, which is one of the acrylamides, you end up with a thicker adhesive layer combined with the tags, and that was the group that provided the best results. So it's not that simple, right? You can't just use the acrylamides. There's some tweaking that needs to happen for a good formulation. The other thing that was interesting about this work is that she was able to develop a technique in our lab to do 2D mapping of the adhesive interface with our IR microscope. So this is in mid-IR. I mean, we've seen this in Raman microscopy being done before, but this is, I think, one of the first times that I've seen this done in mid-IR. Okay, so I'll stop that. That's the preamble of what has happened in the past and some of the things that we are still excited about. But changing gears now to the biofilm and how that biofilm contributes to those mechanisms for hydrolytic degradation. We're going to start with the biofilm, right? Because I always get myself confused. The biofilm and the MMP, the substrate. So I'm actually going to talk about the MMPs before the biofilms because that kind of ties in a little bit with the acrylamides. So this is also work that was developed here that started when Anna was in the lab. We were writing that paper on the acrylamides and trying to figure out how inhibitions were working. That's when we came up with this NNGH inhibitors, and Poliana did a bunch of work with that. Our proposal with this is to use this inhibitor, which is known, it is an MMP inhibitor. It's already known to be an MMP inhibitor and used as a cancer drug, basically, so to prevent metastasis. So we thought, what if we use that? We could combine that and use this to preserve collagen in the adhesive layer. So Poliana did a lot of seminal studies on this. I showed you the hydroxyproline assay before, and I was pointing out the acrylamides before, but the NNGH is here, right? So through different mechanisms, right, the acrylamides provide protection through those supramolecular interactions that I was talking about, but the NNGH actually, you can see from the gel zymography, completely inhibits the activity of MMPs. And in situ zymography, when Poli did that also, you can see that compared to the control, there's a lot less activity, MMP activity in the presence of NNGH. The interesting thing about this, though, is that this is a nanomolar scale inhibitor. So you need a very, very small concentration in solution. So there's a big caveat to that for you to inhibit the enzyme. So our proposal now is to try to immobilize this and actually use this in a material. So Fernanda came back as a postdoc, and when she started, this is actually, we're very proud of her. This is the work that earned her the Marshall's postdoctoral award at the Academy of Nanomaterials. And she and Matt actually did a lot of work on the enzyme kinetics of NNGH, and you're seeing several different derivatives of that NNGH. And basically what we're trying to do is look at different chain length extenders. I don't think I have the structures on the slide for a good reason. But long story short, with all of this, is that you can see that the enzyme activity, either with the parent molecule or all the other hydroxamic acid versions of it, there's a very sharp decrease in activity when you pass a certain concentration. I mean, again, not going to go dwell into details into this, but the take-home message with this is doing the modifications that we did not decrease the activity of the NNGH. This is another hypothesis that shows that even when the hydroxamic acid in the NNGH gets hydrolyzed, even though the inhibitory activity drops, there's still some inhibitory activity, and this is what we kind of show in the in situ zymography. I mean, I'm showing you those results because qualitatively there are some groups where you can see that there's less activity, but this is I think something else we have to come to terms with in dentistry. This is a difficult test for you to do quantitative analysis on, right? So, we're still trying to follow that, but we have evidence in other tests that demonstrate that it's a very, very potent inhibition of MMPs. Then what she did also was a storage modulus. This is a very busy slide, so bear with me here for a minute. She did the same storage modulus, shear modulus, that experiment that Poli had done, where she showed with the acrylamides that there was an actually increase in the modulus over time with the acrylamides, again, supramolecular interactions. And we were not able to observe that same effect in collagen in the presence of NNGH. So I think this is where we are still working on this project, because even though it has a very strong inhibition potential in solution, once you put that in the substrate with all the other proteoglycans, with everything else that goes on, it becomes a lot more complicated. So one thing that we need to do, and again, these are not tethered to anything yet. Those are all the variations that I showed you, and they're not methacrylated. So the next step for this is to either do the methacrylation or, develop a collaboration with Anna that developed all this drug delivery systems for potentially using this in a primer layer. We've been able to show, though, that there's a trend. I wouldn't call this is not something I'll be jumping up and down like the other ones that I showed you before, but there is a trend for better preservation of microtensile bond strength with those with the presence of NNGH compared to the control. So basically the control here, or no treatment, is the only one where you see the letter B. So that's the only one where there was a drop between water and biofilm storage of those microtensile bond strength sticks. And for all the other materials, there wasn't a drop. But when you compare all the orange bars and when you compare all the blue bars, water and biofilm, they're all the same statistically. So that's why I'm saying, there's a trend, but I wouldn't call that like life-changing at this point. And then Fei did again, she repeated the hydroxyproline assay with chlorhexidine as one of the comparisons, and 2% chlorhexidine, so that's a boatload of chlorhexidine, has the same effect of 100 micromolar of NNGH. The other thing that she did is then she looked at collagen degradation, but just discs of collagen that she made out of red tendon tail collagen. And the pictures are kind of misleading because in here you see that the NNGH, it seems like the control looks like it has larger pores, right? And then smaller pores here. But you have to remember that this disc was just a teeny tiny remnant of that disc that was on the side of the well plate, whereas in this ones, the disc were actually can actually take the disc out of the well. So the images are just to show details of the collagen network, but you are seeing that there's a huge improvement on the preservation of the collagen itself. Okay, now we're going to move to the biofilm. So the challenge with the biofilm, when you look at this, and you'll see a lot of, this is not new, right? That people are trying to decrease the biofilm formation on the surface of nanomaterials, right? I mean, this we've known forever how caries forms and how secondary caries had to do with some biofilm that's forming on the material. I think what's new about where the field is going, and hopefully where our lab is going along with the field, is looking at the biofilm as a more complex structure than just focusing on S mutants. Because S mutants, I hope everybody in the audience is familiar with that. Bacteria, we know that it's the most virulent and one of the culprits basically for caries formation because it's acidogenic and aciduric. But there's more complex interactions in the biofilm in the environment with commensal bacteria and micro. So this is what this figure here is from a work from Michel Kuh is showing, that there's different pockets within the biofilm where the composition of the biofilm itself is very different. So our idea here is not to completely eliminate the biofilm. We wanted to modulate the ecology of the biofilm in such a way that you would actually preserve the commensals and empower the commensals to get rid of the virulent bacteria. So I know this sounds kind of up in the air, Age of Aquarius thing, but we're going to go over the details on how we're planning on doing that, and hopefully that will make more sense. So just to set the stage again, there's been different approaches to try to combat those biofilms. One of them, ranging from surface modification, so the quam materials where you have quaternary ammonium moieties at the surface is a very well-studied one, where you basically have a cationic charge at the surface of the material that, I'm not going to go into the mechanism, but that eliminates the bacteria from the surface. There's other people who have looked at, so our neighbors here, between Adriana and me, the University of Washington people in the chemical engineering department, they came up with a carboxybethane type of chemistry that switches between killing and antifouling. So there's all sorts of things that can be done to the surface. Other things have been in the realm of nanoparticles and small molecule agents. You can actually see a bunch of those are for drug delivery. Michel Kuh has a lot of work into this. And in this case, some people have been focusing on the EPS degradation rather than killing the bacteria. So EPS being the extrapolysaccharide matrix. That's the glue that keeps the biofilm as a cohesive group. So rather than killing the bacteria, some people have, myself included, have proposed to disrupt the production of that glue rather than killing the biofilm. And then there's other people who are looking at different types of physical removal. Michel Kuh, again, has a very, so the Nature Reviews is where this paper came from, but he also has another paper where he described the micro robots basically that go onto the surface of your tooth and kind of scrub the whole surface around. So anyways, if you don't follow him yet, please, he's the bomb in all of these things. So what I'm going to talk about is EPS degradation and how we can figure out a way of decreasing the production of that EPS. And the work that we're doing is building upon this work from the group that worked with Hui Wu, who is now our associate dean for research here at OHSU, who's a very accomplished oral microbiologist. And this is their work that I'm showing here. He came up with this molecule that I'm going to call from now on the G43. So this is the structure right there. And he did a lot of molecular docking and molecular modeling, computational modeling of that molecule to demonstrate that it had interaction with the pocket, with the actual active site pocket of GTF, the glycosyltransferase, that actually produces those glucans that lead to the formation of the EPS. Are you following me? So what he was able to show is that this molecule here stops the enzyme that makes the glue. Not only that, it only does that for the bacteria, for S mutants, and not for the commensals. So this is one of the first works that has shown that there's a small molecule that targets specifically the virulent bacteria and not the commensals. I'm not going to go into a lot of detail into this. That's why please check out the reference if you're more interested in this. I know this is being recorded, so you can go back to this and look. These graphs are showing the relationship when you have a dual species biofilm. So when you're growing, say, S. gordonii and S. mutants together, how the populations change based on the concentration and the presence of G43. So the main conclusion of this paper is to show that the G43 only inhibits the S mutants and prevents that from being enriched in the biofilm. So the idea with this is if you have a commensal that produces hydrogen peroxide, for example, that hydrogen peroxide, that increased hydrogen peroxide concentration is toxic to S mutants. It's pretty cool, right? Pretty clever way of doing that. Because we are materials people, and I say that I'm a materials person that has been contaminated by the biofilm microbiologist that exists here at OHSU, but because we are biomaterials people, what we're trying to do is modify then those molecules to then try to decorate surfaces with those molecules so they can be available, that you don't have to worry about them being delivered onto the site. So Alex Candle, who was a postdoc, research associate here in the lab, who is now at PNNL, the Pacific Northwest National Lab, also up the road here on I-5. When he was here, he designed and synthesized all those molecules that you're seeing on the screen. And then our goal with all of this is to have a side chain extender that would have a methacrylate. So then the G43 portion could be, exposed to the environment, and then you're inhibiting those enzymes at the surface. I mean, there's lots of discussion onto this, whether this is a good idea or not. That would take more than the time that I have here. I'm happy to take questions. Wink, wink. I'm telling you, this is a good question to ask. So Polly also, when she was here, she did, so again, I'm just gonna say, our objective with this was not for this molecule to be bactericidal. We didn't want to kill the bacteria. We want to just eliminate the bacteria from the surface, or at least disrupt the biofilm. So what Polly did was she tested the control, like wild type S mutants, and then no treatment, versus a complete knockout of GTFs that Hui gave us. So this is what I'm going to call Delta GTF B, C, and D. So this bug is genetically modified, so it doesn't make any GTF, any GTF, so there's no EPS formation, and you see that there's no biofilm that can form if that happens. Then we looked at the parent G43 and all the different variations that Alex made. And you can see here, this is, I think, a nice picture because it shows that, the bacteria is not completely eliminated, but the biofilm is totally messed up, right? It's a lot easier to remove. And we found one of ours, at least one or two of ours, that had the same effect as the parent molecule, albeit at a slightly lower, higher concentration. The other thing that Polly was able to show is that the luminescence, or in this case, we're looking at the bacterial viability, that did not change. So we were not killing the bacteria, we were just disrupting the biofilm. And then when you look at the water-insoluble EPS, so how much EPS is being formed, again, triple knockout has nothing. And then some of our variations had the same reduction in EPS formation as the parent molecule. So we're pretty excited about that. So we're following this guy here. All right, so then Polly, and you'll see a picture of Yuji in a minute, she also decided to look at how the mechanism, how that G43 modified molecule was affecting the glycosyltransferase. And you have to remember that that enzyme has actually two different functions. It both breaks down complex sugars into the monomers to make the glucans and also synthesizes the glucans. So it has a hydrolase and a transferase activity. And what Poli was able to show is that that enzyme, I'm going to go through this a little faster because I'm looking at the time. What Poli was able to show is that this enzyme gets completely inhibited by G43 or gets significantly inhibited by both G43 and G43 tag. Poli is very talented, and she did that both with the recombinant enzyme that Hui's lab provided, and also with enzymes that she extracted from the actual biofilm that we grew. Then Yuji came up with this way of measuring the kinetics of inhibition, and this is what we're calling from now on the PNPG assay. So this is basically a fluorophore that has a glycosidic linkage, and in the presence of the enzyme, that linkage is broken, releases this para-nitrophenol that has a color. So we basically do a colorimetric assay to look at the enzyme activity. And the take-home message from those two graphs that are here on the bottom are that the enzyme activity as a function of the substrate concentration, so this is how much PNPG we're putting into the medium, decreases in relation to the control. So this is the DMSO control, and this is the enzyme. This is the G43 treated enzyme for both GTFP and GTFC. So the only issue that we have with that is that the concentration at which that happens is 50 micromolar. And that's a very high concentration in the drug development world. So that prompted us to actually go look for other molecules. And that's where Fernanda comes in again. You will see work from her again. So we then learned more, well, and years have passed. So then the molecular dynamics, docking and all of that has improved. The technology itself has improved. So what we decided to do is to say, okay, rather than having to develop something de novo, why don't we look at a library of drugs that are FDA approved and see if they can be effective against our enzyme of interest? So this is what she did. She screened. This is a video, but I don't have to play that. She basically screened 16,000, no, not 1,600 molecules to begin with, because otherwise that would take forever. And, for docking into the active site. And then from that in silico screening, that was over 1,600 molecules, we did then some biological screening. So the workflow is this. She would take that library of drugs, do the in silico docking into both in the active site and then what we call blind docking that's around the entire protein. So you can have some what we call allosteric interactions. Select a few candidates from that and then test those candidates for those three things that are on the screen. So we wanted to see if this would, one, inhibit PNPG, the enzyme activity in the PNPG test that I just showed you, if it would reduce the biomass or at least modify the morphology of the biofilm and not reduce viability. So there are three tests that we did. We did the enzyme activity, and then we did biofilm screening of 57 drugs that came from that 1,600 that she started with. I am not going to go through this. I just want to tell you that this is a lot of work to get to the next slide that I'm going to show you. So this is color coded with the red being drugs that do not eliminate biofilm, yellow being the ones that eliminate biofilm but are also decreasing viability, which means they're likely toxic. And then green are the ones that eliminate the biofilm without reducing viability at the concentrations that you see on the screen, 50, 25, 12.5, and 6.75 micromolar, so a twofold dilution. And you can see that this guy right here eliminates the biofilm, but eliminates it, like it really annihilates it. And I'll show you some other tests that we did in later on that shows that this drug basically reduces viability. Long story short, we were able to develop or to identify first this one drug, then we found another one that's not on the slide here. One drug that does eliminate the biofilm up, oh, this is actually switched. I apologize. This is actually switched. This should be here, and the 50 micromolar should be here. My apologies. The 50 should be on the bottom. So at 50 micromolar, that drug, the 3.8, completely eliminates the biofilm. But as you decrease the concentration, even at a lower concentration, the 675, there's some disruption of that biofilm. So this is what we were interested in. We didn't want it to be, even if it didn't eliminate, we know that because this is happening, there's something happening to the architecture of the biofilm. So again, then Fernanda concentrated or focused on those 3 drugs. We didn't want to eliminate or not test this number 2 or NBC, as we're calling it from now on, because it eliminated the biofilm. So we wanted to continue investigating that. Even if it wasn't through the mechanism of inhibiting enzyme activity, if it wasn't toxic to other cells, maybe it could be an interesting candidate. So then 3.8 and 6.4 were the other two drugs, and radatinib and pranlocast were the other 2 drugs. And what you see here in this cartoon, basically representation, is what I just showed in the previous slide. The only one that had the all green check marks was this guy here, 3.8. So then we did, in collaboration with Wei, we did MST binding, which is the same as, it's a different technique, but does the same thing as octet red, which I think people are more used to. And that is just basically a technique that allows you to see where that enzyme is binding, because then you give just the active site and the molecule and check for binding on that active site. So I can confirm that the 3.8 is actually binding to the active site and not to some other part of the molecule. And both those 2, 3.8 and 6.4, did that. So we know, we are happy because that's the we can confirm that that's the mechanism. So you also did what you see here. I'm not going to go into detail. We did octet red with that also, but also the MST. So we checked the binding with 2 different things. And the IC50 here is the minimum concentration that you need to inhibit the enzyme activity in the PNPG assay by 50%. So, the screening is progressing and 3.8 is holding steady. Okay, so then when you do this live dat assay, you see the control, that's DMSO, number 2, everything is red, so it means all the cells are dead. Then 3.8, if you look closely, I mean, it has a lot of green, right? So the cells are not dead, but they're not as organized and not forming a beautiful carpet as the control is. Not only that, when you do a test that we call impingement assay, which is basically taking that sample, and it's a glorified water gun, basically, that you take at a very controlled pressure, you squirt water on that sample. And for samples where it's more difficult to remove the biofilm, you see that there's a halo only where the thing was actually touching. But if you look at the pranlocast, both 3.8 and 3 and 6.4, it's a much more diffuse halo. It's actually even more difficult to measure, which just tells you that this is an easier to remove biofilm. It's not as cohesive of a biofilm as the control. So then when you look at this with a different technique, this is, we're staining the biofilm, the cells with cyto9. So what you see in green here is S mutants, is the nucleus of the cell. And what you see in yellow, this is cascade blue, is what we're staining the EPS with. So you can definitely see when you're doing these images that whereas there's a homogeneous distribution of S mutants and EPS on the control, it's a lot more disorganized in the 3.8. So a lot of the things, I'm going to go quickly onto this because a lot of the things that I'm showing you here are just, validation over validation over validation, that with different tests we're showing the same thing. This is a different rendering, different image rendering. I just like those pictures. I think it's actually good to show it. This is possible to do. From this, we can do this biofilm rendering that shows the 3D structure of the biofilm, and a heat map of where both the bacteria and the EPS are distributed. So the conclusion is the same. We're disorganizing the biofilm. And this is, I'm gonna skip over this because I see it's 9:44 already, but this is what this is showing. This is the quantified data from this. Right, so we took that image, we did the 3D rendering, and then we quantified the amount of blue and green that you have, and you come up with these graphs. The conclusion being that we have less EPS with the drug than in the control, and the bacteria still survive. All right, so this is great, but what I told you in the beginning is that I wanted to mess up with the ecology of the biofilm, not that I wanted to kill or disrupt S mutants. So the next part of the work, and this is basically the last part of what I'm going to talk about. Let me just check on something real quick. Yeah, great. I'm gonna have time. I have 10 more slides to go. So
Don't rush. Take your time.
Yeah, well, I want to be respectful of everybody's I hate being late. A Virgo person in me, like, all right. Anyways, so I told you that I wanted to mess with the ecology. I did not want to just kill S mutants or eliminate S mutants. So what we are doing now is what we call a reductionist model. I mean, our overall goal is to eventually go to a microcosm model, which we kind of did, but I'm not showing the results here. But to mechanistically study what the interactions are between our bacteria of interest, we went to a reductionist model. That just means we tested S mutants along with S gordonii or S sanguinis. There's things for both of them in the next few slides. And the reason for that is also on the screen. I kind of alluded to that when I first explained this, is if you give an advantage to the commensal bacteria, they're competing for binding sites on the surface of the enamel. So that actually gives less opportunity for the S mutants to attach. The counter argument to that is that S mutants also attaches to S gordonii, right? So because they have some synergistic relationships. But the idea is if you have high enough of a concentration of the gordonii, then number two will happen, that you'll have such a much higher concentration of hydrogen peroxide, which is inhospitable for S mutants. So to close that loop, because this is all a complex interaction, S mutants also has some protective mechanisms and produces this bactericin that can actually eliminate that it is not sensitive to, but S gordonii is. So we went into this because it's not enough just to look at the S mutants, because there's a lot of protective things from Vionella parvula, for example, is the next one that we're going to look at. Okay, so this slide, I have to think because they're so complex, stay with me here for a minute. What you're seeing on the left side, those different circles, is just looking at the biomass with crystal violet for either DMSO on the top. Each one of those has a top and a bottom of DMSO versus the drug. It's always 3.8. But what you see on the bottom here is the type of bacteria you were testing. Okay, so this one is UA159, so it's wild type S mutants. This is a knockout for GTFB. So this one does not produce GTFB. This is a knockout for GTFC. So there's no GTFC present in that material or in that culture. And again, thank you, Hui and Hua Zhang, for providing us with those strains. Then this is wild type S mutants with wild type S gordonii. So this DL1 is the wild type. These are strains that you can commercially obtain. Then we got the gordonii with the delta GTFB, so the knockout of GTFB and the knockout of GTFC. And we did that because our hypothesis was that in the presence of 3.8, the biofilm would behave the same way as the GTFC knockout. So that was the hypothesis driven why we're doing this experiment, right? Wouldn't that be great if the drug is actually doing the same thing as the GTF not being there at all? So that would be the ultimate test, right? All right, so the summary. So this is how I got the data. The left-hand side is how I got the data. The right-hand side is the interpretation of the data. So this on the y-axis is showing the biomass, and then here the different types and combinations of what type of biofilm I'm looking at. The blue line is the results that you got with DMSO, so just the control, and the green line is what you get with the drug, right? So this is making sense of that. I'm showing you the data and then trying to make sense of it. So the first thing that stands out is that when you don't have GTFC, whether you have drug or you don't have drug, there's a decrease in the biofilm, right? That's because there's no APS to be formed, and the biofilm reduces. The other thing that you can see is that the green line is always below the blue line. So having the drug in there, even though this is not statistically different, having the drug in there decreases the biomass of the biofilm. And having the knockout of the GTFB doesn't change much. So this is also showing that GTFC is the enzyme we should be targeting, not the GTFB, because you see wild type and GTFB, they have the same basically on the control. Having the drug in this wild type decreases the biofilm a little bit more than the wild type, but in the control group or no drug, GTFB knockout behaves more or less the same as the wild type. This is complicated, so I have to stop and think about what I just said. Please ask questions if you have them. Now, what we would like to show, though, is what is that we're trying to accomplish here, is to show that the wild type with the drug, so this is what this comparison is. If you look at wild type versus this is wild type with GTFC. This has to be wrong. Oh, this is the 3.8. So this is the bottom, not the control, but what you see with the drug in the wild type versus the drug with the DMSO sorry, I'm going to say this again. The part that is confusing is this, and this is this. So this is showing the wild type with the drug, and this is showing the GTFC knockout without the drug. And when you quantify the biomass for those two things, so this comparing this guy with this guy, this is what you get. They're statistically the same. So we were screaming. These are the types of results that we scream about in this lab. We're very excited to see that. Then we did that 16S sequencing, because what I didn't tell you is this is the total overall biomass. So I'm not differentiating between one type of microorganism versus the other. Then we did RNA-seq with all of this using nanopore sequencing analysis. So big shout out to Jonathan Baker, one of the microbiologists in our cluster, microbiology cluster here, that the S mutant, this is what this 16S shows, is that this S mutants is preferentially eliminated from the biofilm in the presence of 3.8 or absence of GTFC, which is what this is showing here. Something else I need to tell you is that we started out, well, I'm not going to go into all of that, because then it gets the composition of the biofilm that was initially. The bottom line with all of this is that we've been able to demonstrate that using 3.8 with those biofilms has a similar effect of knocking out completely the GTFC. All right, so this is the conclusion on that. The one project that I'm going to have very few data to show you, because it's just starting in the lab, is one that builds off of the work from Conrado Aparicio, who used to be in Minnesota, is now back in Barcelona. And he has developed those different peptides that are antimicrobial. And just like what I am saying, what he was trying to do is to immobilize those on the surface, right? So the same idea that I was talking about. We try to modify the materials, either the adhesive layer or the composite itself. And it started with, so the description that you have here on the right side is understanding the different peptides and how the acquired pellicle works, that Musa and Siqueira, Walter Siqueira, and the group from Walter Siqueira, our friend from Saskatchewan, they're in Canada. A few years back, they have this very, very nice review that shows all the peptides. So that's why I brought the figure here. It's very didactic. If anybody's interested in this, this is a good first read on that. Anyways, and what Conrado was showing was that with that peptide, along with Alex Falk also, is that you can inhibit the biofilm formation on the surface of the material. So now, Fernanda Suzuki her project now is, what we're trying to do is, what if we could, instead of having a new peptide, we could trick the surface, or modify the surface in such a way that we can favor some of those peptides that Walter has described that already have some antimicrobial effect, and we can enrich the surface with those peptides that are already in the saliva, so you don't have to do anything with the material. So what if we could change the surface in a way that we can attract those antimicrobial peptides to the surface? All right, so we had to start somewhere. We had to develop a bunch of methods that we didn't have in the lab. So what Fernanda did was, looking at bookends of things that could be different on the surface of materials. So one of them is hydrophobicity. So that's the easy one to test, is either a super hydrophobic or more hydrophobic surfaces versus a hydrophilic one. So she worked with discs with bis EMA and discs with PEG DMA, so hydrophobic to hydrophilic. And then we did proteomic analysis. So actually, Yuji was here when we started doing that. We did proteomic analysis of those discs that were immersed in saliva, like whole saliva that we collected from volunteers that basically are all the people in the lab, spit on a tube. And we have an IRB protocol for this. Don't get me wrong, but we collected that saliva and then looked at what attaches to the surface just based on those two very, very simple things. So in here, I'm not modifying the surface at all. I'm just saying that we're looking at the bookends and developing methods. But actually, to be honest with you, I just did it as a pilot because, again, I wanted to figure out how to measure these things. But it was very interesting that in PEG-DMA, we actually saw much more protein attached. Actually, that was not surprising. We saw much more protein attached to the PEG-DMA than to the BZMA. So we saw that with ATR, and look at the amide-1 bond. We also saw that the PEG-DMA, the type of protein that attaches to the PEG-DMA, is more similar to saliva than BCMA is. And on top of it, it was enriched with alpha amylase and using MUC5B. And it just so happens that those two enzymes there, or those two proteins in there, are anchors, or they can be anchors for commensals more than virulent bacteria. No, I'm not proposing that we all restore teeth with PEG-DMA. I'm just saying that with a very simple change in that surface, right? In that surface, we were able to show that there was, in fact, a difference in the types of proteins that attach to the surface. That had been shown for differences between metal and polymers, or ceramic and polymers, or ceramic and metals, but not for oral saliva. I mean, there's no other type of saliva. Not for any oral fluid, right? In a same class of material. So the only change that we have here is the hydrophobicity and hydrophilicity. They're all methacrylates. They're all dimethacrylates, right? They have TG above room temperature. So that was, I mean, okay, is this a fluke? Can we show this again? Anyway, so then she kept on going with this, and we did biofilm biomass on top of those specimens, and you see BZMA, and this is the 50-50. I forgot to say, we did 100% BZMA, 100% PEG-DMA, and then the 50-50. And you see that you have lower biofilm formation on the PEG-DMA. And interestingly, too, is you see that in the BCMA, you have an enrichment of S mutants. So this again is you take the saliva. We took the saliva and look at the proteins. That's the first part, right? And then I'm showing you that PEG-DMA has enrichment of alpha amylase that S. gordonia, for example, loves. Then I'm showing you here that when you took the saliva, the biofilm that came with the saliva from the donors, and you look at the actual species, so you actually do microcosm analysis and you look at what species are binding, there's a lot more S mutants binding in the BCMA. Now, is this because we have less amylase, or is it because it has more biofilm altogether? So I don't know the answer to that. That's what we're working to find out. So that's, stay tuned. That's coming up next in the lab is work on that project. Then the last thing I want to talk about really quickly, because I think this is very interesting, is we kind of pivoted in the last couple of years to try to use some of those technologies that we developed for adhesives for other biomedical applications. So think about bone and dentin. They're very similar, right? In both chemical composition and structure, right? See, if you think about trabecular bone versus cortical bone and the different densities in the bone. So Sam Weber, who was also a mechanical engineer, not in our lab anymore, he tested, and this is all commercial materials. I didn't do anything with the acrylamides. This is all commercial materials. Again, using the bioreactor. Rafael from Odemi says he loves this picture because there's 3 different products from Odemi in the same picture. Again, I have a conflict of interest for this, but not for this, okay? So I'm not getting money for this. But anyways, Sam used those pieces of equipment to actually have a piece of the bone embedded in epoxy that we then cemented with a regular bone cement. Like that's basically PMMA. If you think about it, it's the same thing as we use for denture bases. And then he tested that in the bioreactor. Then we looked at shear strength between a titanium puck that was bonded to the tooth that way. So doing the standard of care, which is no adhesive at all. This is how much bond strength you have, and you put adhesive, any adhesive, even our bad adhesives, you get between 10 and 20-fold increase in the bond strength. Not only that, the same mapping, this is Fernando Suzuki that did that same mapping. You see that there's a lot more interaction between the cement and whatever material and the bone than without the adhesive, obviously, right? Because the adhesive has lower viscosity, so it penetrates the nooks and crannies. I don't have to explain to a dentist why that works, right? But then talking to the orthopedic surgeons that were working with us, they're going, what? That is what? So this is another one that's interesting, and hopefully we'll be able to do some. There's two things. We started looking at this for hip implants, but there's actually even a better application for this, which is in, say, for example, if you have spine fracture and you have to put plates on the spine, they usually need screws to go into this and stabilize, and the screws need to be long sometimes, and the failures happen at the screw, right, rather than on the plate. So if we can have an adhesive that helps reinforce this and can even if it doesn't eliminate the need for the screw, but it allows you to have a shorter screw or fewer screws, wouldn't that be great? So that there's this is how life goes. There's another student from BME, and Anna forgot to say she's also part of biomedical engineering. There's this other student who is doing that work exactly with plates, and he has an animal model for that already. So stay tuned for that. Now, shameless plug for the biomaterial synthesis and characterization core. Like I told you, Steve is the boss of all of that. All the stuff that I showed in that presentation is available through the core. So if anybody wants to collaborate, I would love to collaborate in an actual scientific capacity, but if you just want us to send us specimens to test, please scan the QR code. Steve's gonna be happy. And with that, I want to thank the people who actually did all the work. I get to look really good in these presentations because of the work that they do. I'm not going to say their names because the list is vast. Thankfully, we're being fortunate with recruiting. This is our annual Pfeifer Lab picture at Research Day here at OHSU. This is a nice one when Enrico left. This is another PhD student who was here. And there's a few people who are not from the group. So like this is from he's from Jonathan Baker's lab. This is from Luis Bertosoni's lab. And also I would be remiss if I didn't thank the funding sources. NIH and IDCR has been very good to me throughout my career. All the people who are listed and shown in this picture, including Ray Bowen, who started this whole thing with BizGMA, and I was lucky enough to take a picture with him. Justin Merritt, the person who started all the microbiology, was the patient zero, basically, the first microbiologist in the department. Jeff Stansbury, my postdoc mentor, without whom I wouldn't know half of what I know about polymer chemistry. Anna, wonderful person, without whom we wouldn't have a lot of the papers from the time she was here. And also the graduate program from the Piracicaba School of Dentistry, who sends me. I feel like I have a branch of the school here in my lab. They're very, very good collaborators, and also my kids who